Go ahead and get started. I'll give a little introduction. Good morning, everybody that's online with us. I appreciate your tuning into this. Today we're very happy and honored to have Dr. Bettina Fries to give a talk to us on COVID-19 and the history of pandemics. Dr. Fries is the Chief of Division of Infectious Disease and Professor of Medicine, Molecular Genetics, and Microbiology at the Renaissance School of Medicine at Stony Brook University. And it's being brought to you by Brookhaven Women in Science. And Brookhaven Women in Science is a diverse and inclusive community that promotes equal opportunities and advancement for women in world-class science. So we're very happy to have Dr. Fries here with us. And with that, I will turn it over to her and let her start sharing her screen and give us the information about COVID-19. If you have questions, and I hope you do, please give them into the chat, type them into the chat, and at the end we will read the questions, or I think if Dr. Fries can see them, I'm not sure if she'll be able to see them or not. If not, she can go ahead and answer them. If not, I will read them. So, Dr. Fries, it's all up to you.
Okay. Does everybody see my slides?
Yes.
Okay. Yep. All right. So I'm going to talk about COVID-19 and about the SARS coronavirus 2, which is a novel emerging pathogen. So I have no conflict of interest to declare. And the objective of my talk is going to be to give you some background information about this virus and to sort of explain how this pandemic is different from other pandemics and to discuss the challenges that we face managing this pandemic, to sort of answer questions like how did this pandemic evolve, how does this pandemic compare with others, what did we already know and what did we have to learn? How did we get organized during the pandemic? What were the challenges? I'll talk a little bit about what's happening now and what continues to be a challenge. So this is a new virus, and the official name initially was called 219 novel coronavirus, and then they renamed it SARS coronavirus 2, and that's what it runs by now. The disease is called COVID-19. The virus is called SARS coronavirus 2. Early on, you know, nowadays when you have a new emerging pathogen, it's actually pretty easy to figure out what this pathogen, where this pathogen is coming from and who it's related to. So early on, they published a paper showing 10 genome sequences of patients that presented with a disease. And based on these genome sequences, they found that this novel coronavirus had 88% identity with two bat-derived SARS-like coronaviruses, which have these funky names here. And both of these bat coronaviruses were collected in eastern China. It had 79% identity with SARS coronavirus, which is a virus that caused an outbreak in 2013 in China. And it had about 50% identity with the MERS coronavirus. So the SARS coronavirus, the MERS coronavirus, are both two coronaviruses that have the SARS coronavirus caused an outbreak in 2013. The MERS coronavirus intermittently causes small outbreaks, predominantly in the Arabian Peninsula. There are 4 common coronaviruses that cause cold-like syndromes that we consistently see every year in our patient population. And these coronaviruses are diagnosed by our respiratory viral panel, which is a PCR-based viral panel that we do on anybody who comes into the ER and has an upper respiratory virus. So this novel coronavirus falls within the subgenus of the beta coronavirus's relatively long branched length to its relatives, the bat viruses. And they make all these nice phylogenetic trees, and based on that, you can basically, am I on the, you can basically look at the relateness of the virus. So these coronaviruses are called coronaviruses because they have this crown-like structure. They have this very prominent proteins on the outside sticking out. And if you look at this under the electron microscopy, it basically gives you this, view that looks like a crown. And in the good old days, the only thing we really had was good electron microscopy. And based on good electron microscopy, we could identify the shape of a virus, and based on that, we could put the virus in relation to other viruses, because the shapes are very classic in some viruses. So on the outside of this virus are 4 proteins. The S protein is the most dominant one. It's the spike protein, and it's talked about a lot. The M protein and the HE protein all are expressed in smaller quantities. The spike protein is important because the spike protein is actually very important in the pathogenesis of the virus, as it is the protein that binds to the cellular receptor that allows the virus to invade into the cell. So all mammalian viruses, and I don't know, but I'm speaking to a virologist here, you may already know that, but all mammalian viruses need a receptor to enter a mammalian cell. It makes them different from plant viruses, who they can get into a cell in a different way, but a mammalian virus needs to bind to a surface structure, and the process of getting into the cell is an active process. And so it's always very important when you are dealing with a virus to figure out what that surface structure is, what the receptor is, because you're going to try to see whether you can actually use this as a target for making antibodies, for instance. And of course, we are born without the need to be infected by these viruses. So these viruses also have been around much longer than us. And so they bind to surface structures that are usually used by our cells for something else. And a lot of times these surface structures are very important molecules on the surface. So you can't just eliminate them or neutralize them. So the SARS virus, the thinking is that it binds to the ACE receptor, and that's the angiotensin receptor, and I'll talk a little bit more about that. It then needs to be modified and is then actively taken up. So this spike protein, the reason why we sort of knew that it was binding to the, or why we suspected that it was binding to the ACE receptor is because a comparison of alpha and beta coronaviruses identified 2 notable genomic features, and based on structural studies and biochemical experiments, it appeared that the spike protein was actually very similar to the spike protein of the coronavirus that caused the SARS outbreak. And for that coronavirus, we had already established that the ACE receptor is actually the receptor that allows the virus to enter the cell. So this is the receptor binding unit of this spike protein, and you can see that they sequenced it very carefully, and they compared it to a bunch of other coronaviruses, some of them derived from bats, and some of them derived from other intermediate hosts, one of them being pangolin, and I'll talk a little bit about that. And they found that in this spike protein is divided into sort of different different portions. There's an S1 subunit, an S2 subunit, and there is a subunit which is referred to as the receptor binding domain. And that basically is sequenced. And based on that, you can see that that it's the most variable in 6 amino acids, and they have been shown to be critical for binding to the ACE2 receptor, and they basically determine the host range. So because viruses have to bind to receptors, they also then become very host specific, because these surface receptors are usually very sequence specific for individual hosts. So even though other hosts have AC receptor-like proteins, they are not identical, and that's why viruses can bind with a higher affinity to certain hosts than to other hosts. And that host range is actually very important. And that's also very important from a pandemic point of view, because if you have a virus that can infect everybody, then it's virtually impossible to ever get rid of this virus. Some viruses that can only infect mammalian cells, once you have a very successful vaccine, you can eradicate these viruses, because once you protect the human host and the virus has no host to infect, the virus basically dies, because viruses always need a host to infect. They are almost like parasites. They cannot live on their own. They need to hijack a host in order to use their transcription machinery to basically make their own proteins. Okay, so based on these sequence variabilities, we knew that there were 6 amino acid changes, and these 6 amino acid changes were sort of very unique to this virus, but they were also found in a virus that infects this intermediate host, which is this pangolin. On the basis of structural studies and biochemical experiments, these were very critical, and they determine the affinity of this virus for humans, ferrets, cats, and other species. The other thing that was very important in the spike protein is a polybasic cleavage site, and this actually is unique to this specific coronavirus and different from the other coronaviruses. And these involved 5 amino acids, and this polybasic cleavage site also had certain glycosylations associated with it. Now, this polybasic cleavage site is also not present in the spike protein that of the coronavirus that infects this pangolin, which is thought to be the intermediate host of this virus. So that brings us to a sort of very important understanding. Coronaviruses are viruses that infect animals and humans. And so they jump between animals and humans, and that's what happened in this pandemic. So a virus that is abundant in animals, all of a sudden infected humans, adapted to the human niche, and now very successfully is transmitted from human to human. And that's really the critical event that caused this pandemic. This virus was around before, and this virus basically made a jump in species. Now, when we compare this virus to the other 2 viruses that can cause outbreaks, the MERS virus and the SARS virus, what we knew about these 2 viruses, that they are bad, and that when they infect humans, that they lead to a lot of mortality. So in the SARS outbreak, we observed mortalities in the range of 10%. And in MERS outbreaks, which have been only very few, luckily, we have observed mortalities up to 40% of patients that present with that disease. Now, this and both of these, all three of these, cause these severe upper respiratory infections, bilateral pneumonias, leading to an acute respiratory distress syndrome, horrible changes in the lungs that basically impair oxygenation. These two are related to each other much closer than this one. This is basically also a virus that binds to a different surface receptor, binds to a receptor called CD26. This one, the SARS virus, also binds to AC2. And the outbreak, the first time the outbreak was observed for MERS was in 2012. It's thought that this virus usually lives in camels, and that people that present with these infections are usually intimately, in vicinity of these camels, and basically every once in a while the virus that usually infects the camel jumps species, infects humans, but doesn't really greatly adapt in humans to from there on cause huge outbreak in other humans. But it does happen every once in a while. And it happened, for instance, with a patient in Korea that had returned from Saudi Arabia and then was admitted to some hospital in Korea. He had acquired MERS in Saudi Arabia, and it ended up infecting 120 patients in that hospital. So in that patient, the virus very successfully infected that patient, and that patient then spread the virus to other patients. They caught the outbreak, and the outbreak was contained and didn't spread from there and didn't cause a pandemic. But every time Hajj, so the big pilgrimage in Mecca starts and 2 million people meet in Saudi Arabia, everybody is a little bit scared about this virus causing an outbreak and spreading in that huge, big meeting of people. And so every time when Hajj happens and people return from the Saudi Arabian Peninsula, hospitals throughout the world are aware of this, and this becomes part of our history taking. So when we have patients that present with severe upper respiratory infection, we tend to ask them, have you traveled anywhere? And if they say, yeah, I traveled to Saudi Arabia and I was in Mecca, then we, in theory, if we are good doctors, we know that they could have been exposed to MERS. In SARS, the intermediate host was supposed to be these palm civets and raccoon dogs. These are these mammals that were sold also in these wet markets in China, and they jumped species, they infected humans. The virus then spread from human to human, was successfully contained and nevertheless left China, but left China reeling from it because it really shut down their economy in 2003. And I don't know whether you all remember, but the whole world got scared and everybody started doing conferences on Zoom. So for SARS-CoV-2 virus, initially they didn't know what the intermediate host was. They knew that the virus had originally also come from a bat virus, and that there were bat viruses that were very closely related to this new human virus, but they didn't think that we got infected by bats, but that there was some intermediate host that became the carrier, and the thinking is that it's probably this weird-looking whatever species it is, pangolin it's called. And the reason why they think this is the intermediate host is because it has a very similar receptor binding domain, but it still doesn't have, the virus in this animal still doesn't have this polybasic cleavage site. And they think that this polybasic cleavage site actually then happened through mutation in the human host. They do not think, very different from our president, that this is a laboratory-made virus. And the reason they don't think that is because the virus doesn't have any backbone similar to other viruses. They really think this is a natural virus and that it jumped species, probably jumped into this host, and then from there jumped into humans, and in humans further mutations happened between November and December, and then basically gave us this new human pathogen. So with respect to the clinical presentation, I'm not going to go too much into it, but basically we have patients that present with severe respiratory distress. So one of the hallmarks is that patients that come to the hospital are very hypoxic, and many of them need oxygen. Some of them present in septic shock. Some of them then develop acute kidney injury and multi-organ failure, and that's the reason why we see so much death. They present usually with fever and dry cough and flu-like symptoms. Another hallmark of the disease is that they can have impaired sense of smelling. But as we know now, and we're going to discuss a little later, the clinical presentation can be very variable, and probably 40% of the patients have very minor or no symptoms at all, and they keep running through our communities, and they could potentially infect us all. So one thing to understand is that this disease goes back to something that we already know we have studied in the last 15 years very carefully in infectious disease. And that is this concept of the damage response framework. And what that means is that we need to understand that when we have an infection, that in many cases, it's not the pathogen that does all the damage, but actually the host immune response does the damage. And so there is sort of a fine line between having an immune response that protects you and an immune response that hurts you. And it looks like, depending on genetics, depending on prior exposure, the host can react either way. And so what really drives sort of this curve and understanding when the immune response causes damage and when the immune response causes resolution, a lot of different factors go into this. One is the amount of infectious pathogen we are exposed to, so that's referred to as the inoculum. The other one is a pre-existing immunity that can be sometimes good and sometimes bad. In dengue, it's bad. In dengue, it means if you've seen dengue before, your risk of making bad immune response that is damage-driven is much higher. In coronavirus, that's probably not the case. It looks like if you had exposure to coronavirus before, then you have a good chance of having pre-existing T-cell memory that's actually going to be protective. And then there's genetic predisposition, there's old age, there is comorbidities that can basically contribute to an immune response that's damage promoting. But this is very important to understand, because when we start designing treatment, then we do some of the treatment is going to be targeted to suppress just the virus, just the pathogen, and some of the treatment is going to be targeted to suppress this inflammatory response. So early on, we understood that we were dealing with not a just simple run-of-the-mill upper respiratory infection, but we were dealing with a viral pathogen that was causing significant morbidity and mortality. And we sort of understood from the initial studies from China, and this is a paper published early on in March that looked at their first 72,000 cases. And no matter what our government says about China, on a scientific level, the Chinese physicians very early on shared everything that they knew and everything that they learned on a constant, continuous basis. That helped us. And the Italians did the same, and the Germans and the other countries and the Spanish. Everybody shared in the research community, and there were these big websites where all the data could be placed. And so we really tried to all learn from each other. And that, I think, is a very, very sort of important message to send out there, is that the academicians and the clinicians, they were really trying to overcome all the political noise and really were trying to find solutions. So the Chinese physicians reported that 81% of their cases were mild, and it didn't require hospitalization. But 19% required hospitalization and 14% were labeled as severe. They needed oxygen in many cases. 5% required intubation. They also reported early on that 1,700 of their healthcare providers became infected. And in the healthcare providers, they were seeing very similar numbers. 15% presented with critical and severe disease, and they also saw death in them. And so we, were very concerned about that early on, because, we had to go out and take care of these patients. And it was really, really very important that the healthcare providers didn't get sick, because if they get sick, then who is going to treat the patients? The other thing that became very, very obvious is that the patients that became critically sick and the patients that died were all old. And that with every decade, the percentage of patients dying actually increased. So these were the vulnerable patients. The other thing that was obvious, and this is always very important in the pandemic, because at any given time you have tons of pregnant women running around, and so you want to know whether they are at risk, because if a pregnant woman is at risk, is the baby at risk? And so the sort of comforting knowledge was initially that very different from Zika epidemic, that we didn't think that the unborn babies were at risk. There didn't seem to be vertical transmission. This was based on only 9 pregnant women, but so far this for the most part has panned out. Of course, if you become very critically sick as a pregnant woman, that's not good for the baby either, if you need to be intubated, because that can lead to premature contractions and poor outcome of the pregnancy. So then we had to look at treatment options, and they, of course, had to be evidence-based, and they had to be feasible. And so just to give everybody a feeling, up now we have over 180 trials launched worldwide. Early on, because New York was early in the epidemic in New York and was hit so hard and so with such incredible speed, we didn't really have that much knowledge, and we sort of had to scramble to get clinical trials organized. Most of the data that came from China was uncontrolled, because the Chinese also didn't have clinical trials up and running in the beginning, and they didn't get organized until they really were through the surge, and that's when they started generating controlled clinical trials. So there are 2 forms what happens in a pandemic. One thing is that everybody has an observation, everybody has an opinion, and everybody has experience. And a lot of data comes from that, and some of that data is very valuable, and some of that data is very confusing, and some of that data is very wrong. So the sort of evidence-based dogma of American medicine really required that we get organized and really generate good clinical trials, double-blind controlled, so that we can really go out there and generate data that we can all rely on. Now, I will tell you that part of my job during this pandemic, or a job that I picked and got very involved with, is to get that ball rolling right away, so that we could generate data, and that we could also become a site for clinical trials and participate in this very, very important academic procedure. Now, one of the things that we have all learned is that in a pandemic, the pandemic is so fast that the system is really not fast enough to facilitate this kind of clinical trials. And so by the time you have your protocols written and the IRB approved the protocol, you're already through the surge almost. So you need to be really, really fast. And there's no doubt that our system wasn't fast enough. Other systems were not fast enough either. So, and this was a challenge that, that New York really experienced and learned the very hard way. Other institutions that came after us were a little bit more lucky because they had a little bit more leave time to prepare. The other thing that's very difficult about clinical trials is not only that it's challenging to get IRB approvals fast enough, it's also challenging to implement clinical trials because you're basically going to tell the healthcare provider, the physician that's in the front line, and the patient that's affected, and the family that cannot even visit the patient, that you want to put the patient on a clinical trial, and that the patient has a 50-50% chance of getting either a placebo or the real drug. And that message is not that hard, that easy to sell. It's not easy to sell to the healthcare providers who became completely desperate. It's not easy to sell to the family who commonly had to make the decisions for their loved ones who were intubated in the hospital and they couldn't even see them. And so it's difficult to develop the discipline and really understand that this is very, very important to do. And I think the country who in some ways has done it the best is England, because England, through their national health system, just did a big clinical trial across the entire nation. And that clinical trial gave us very valuable data. And in America, we weren't so good at doing that. We did clinical trials, but we did it on a smaller base. Our clinical trials were also good and also gave us data, but I think that England should be recommended for doing this on a national level. So when you have these trials, and second challenge is, who are you going to recruit? There's an incredible emotional sort of roller coaster going on because every patient is desperate, every healthcare provider is desperate, and every family is desperate. And the press is putting out all of this data that comes. Anything that they hear, they put out. And so every day, you know, there's something new on the market. A lot of it uncontrolled, a lot of it hearsay. Then you have a president who gets into his press briefings and starts putting out all of these potential treatments. And so you get phone call after phone call from patients' families who want their loved ones to go on that drug. And in this arena, you're supposed to do evidence-based medicine. So this can become very, very challenging. And you need to make sure that you recruit in a fair manner so that you don't just recruit the patient of the family that calls you, but you recruit sort of in a random way, because otherwise you have a complete bias in there. And this was happening. Like, I was getting a lot of phone calls of families who were saying, yeah, I know everybody needs to be treated the same way, but my wife is in there and she is a nurse, or my parents are in there and I'm a physician. And so it's very hard to stay unbiased in this arena. And this is something that I have to say, I didn't really anticipate how difficult that can be. And we ended up making decisions that we would generate random lists of who we were going to approach about recruitment into trials in order to stay fair and unbiased. And because otherwise you never recruit the Spanish-speaking patient, because it's so painful to get a translator on board and do all this consent in Spanish. Much easier to do it in English, and much easier to consent somebody with a family already said that they want the patient in there than to go to a family and convince a family that they should go on a clinical trial. But you want unbiased data. You don't want biased data. So the way you have to do it is you have to have a cut through your patient population in order to do it right. And that can be challenging. The other thing that's challenging, even in the pandemic, is who pays for it? Who pays for the person to do all the work? And in our university, we were shut down fairly fast. So we didn't even have people who could help. And we had lots of people who wanted to help, but nobody wanted to go into the room and see the patient, because everybody was afraid to get infected themselves. So the whole recruitment process can become very challenging as well. And then, as I said, you had the press and the social media, and there was a lot of hearsay out there. And so it was sometimes difficult to keep a head on your shoulder and say, okay, we're just going to plow through there and go with evidence-based data. And so there were lots of clinical trials out there, and we also participated in clinical trials. Stony Brook did a clinical trial on convalescent plasma, did a clinical trial on an IL-6 antibody, was early on also in compassionate use trials for remdesivir. And so we were fairly organized and tried to really participate in evidence-based medicine. So in a pandemic, you don't have anything to treat. You don't even know how to treat. So you need to think out of the box. The first thing you're going to try to do is repurpose FDA-approved drugs. Because even in a pandemic, you cannot give patients things that are not FDA-approved. You need a very special permission from the FDA to do that. And that permission is extremely labor intensive, because it involves a lot of administrative work. And that's what was happening with remdesivir, because remdesivir was not FDA approved. So for every patient we were giving remdesivir to, we had to basically contact the FDA and ask for permission. And so the first thing you try to do is repurpose drugs that are already FDA approved. And that's what hydroxychloroquine was, because hydroxychloroquine is FDA approved, not for COVID-19, but it is FDA approved for lupus. And so as a physician, you can give that drug, because as a physician, we do this all the time, that we give a drug that is FDA approved for a different indication. And, but it is FDA approved, and once it's FDA approved, it's up to the physician to basically expand its indication. And so, so we could do it with hydroxychloroquine, but we couldn't do it with remdesivir. And then you're going to look at pipeline late phase drugs. So you're going to call every company in the country and try to put together a list of drugs that is already developed all the way. So all the animals, maybe has already phase 1 data, so you already know it's safe to use it in humans. And then you're going to try to push those drugs into further trials. And that's where remdesivir was stuck at the beginning of the pandemic. It was already, had been already tried for Ebola. So there was already a lot of human data out there. So it was fairly easy to get this drug FDA approved for remdesivir, or at least FDA tested and approved. And then you also look at drugs that are considered elsewhere in the drug. So there's a lot of drugs that are maybe FDA approved in Japan or in Europe, but not in America. That's also a process that you can get rolling in a few weeks. And then you push emergent FDA approval, of course. And then you start the other one, which is you work with drugs that are already out there. So this is the remdesivir. This was a drug that Gilead had developed for Ebola and for Marburg virus infection and for other single-stranded RNA viruses. So it had already been tested in humans. It failed in those trials, but it was out there. And there was very good in vitro data and also some monkey data that suggested it could potentially be efficacious in COVID-19. So Fauci and the NIH took a lead and immediately started an NIH trial. And that trial was started February 21st. They began getting this remdesivir trial, this remdesivir into a phase 3 trial, which is basically a placebo-controlled double-blind trial. And that was led out of Omaha. At the same time, remdesivir was released by the company on a compassionate use. So you had to sign a lot of paperwork for every patient. And we early on got this compassionate use as well. And it was also being tested in a non-double-blind study by other institutions. So in the end, the remdesivir panned out to have some benefit. This is the New England Journal paper. And that basically showed that it shortened time to recovery, and that shortened time to recovery of patients that were randomized to this drug was significant. The median time of recovery was 11 days compared to 15 days in patients that were treated with a placebo. So based on that, remdesivir has received FDA approval. This drug is now used on patients admitted to the hospital, and we use it for 5 days if they are not severely hypoxic, and longer if they are severely hypoxic. And this is standard of therapy. Gilead has now moved to also sell the drug. We also tested IL-6 antibodies, which were produced by Regeneron. These antibodies have not panned out. And there was also an already FDA-approved antibody out there on the market called tocilizumab. These antibodies have not panned out to be beneficial. They don't hurt the patient, but they don't really benefit. Other drugs, lopinavir and ritonavir, that's an HIV drug, also didn't end up being beneficial in controlled studies. And hydroxychloroquine initially looked as if it was beneficial in very small, uncontrolled trials. Once we moved it to bigger trials, and even though they weren't controlled, but they were very big, they were done in veterans, it looked as if the drug was not only not beneficial, but harmful. And we initially had this drug on board at Stony Brook, and we also, within 2 weeks, noticed that the drugs was causing arrhythmias, and we pulled the drug. And the drug was then also recommended by the NIH not to be given on a standard basis. Now, the National Health System in Great Britain, they basically launched a big dexamethasone trial. So looking at, because everybody knew that the inflammatory response in this patient was overwhelming and very harmful, so they initiated a big trial nationwide, recruited 2,000 patients to receive dexamethasone and 4,000 patients to receive usual care. And they used patients that were intubated, and they basically established, patients that were critically ill, and they basically established that among those patients receiving oxygen without invasive ventilation, there was no benefit, but in patients that were either ventilated or received high-dose oxygen, that they had a lower mortality rate if they were treated with dexamethasone. So initially, the NIH guidelines and also the IDSA guidelines recommended not to use steroids in patients on a standard basis. And Stony Brook, very interestingly, our critical care attendings disagreed with that guideline. And we early on used steroids. And I think that's also part of the reason why we actually did quite well in the pandemic, because our critical care people argued that when the patients have overtly inflammatory response, that steroids should be tried. And now using steroids in these patients is standard of care. So this brings us back to the immune response. From the SARS data, we knew that patients make antibodies and that these antibodies can be neutralizing in vitro, that these antibodies last for about 6 months, can be measured in patients for about 6 months. We also knew from the SARS patients that there's a memory T-cell response that is important. And in analyzing the immune response in COVID-19 patients, we have also established that patients make neutralizing antibody responses. Neutralizing antibodies bind to the spike protein, and that patients make memory T-cell responses. And we have also established that some patients that have been exposed to related coronaviruses have pre-existing memory T-cell responses. Although they have never seen this coronavirus, they have cross-reactive pre-existing T-cell memory, and most likely that T-cell memory is the reason why some of these patients actually do better than others, because they basically have a pre-existing immune response. So this is the data from the convalescent SARS serum. So basically they showed that when they used patient serum or rabbit serum from animals or humans infected with SARS, that that serum could basically prevent entry of the virus in vitro. And this spawned a huge nationwide FDA approval of convalescent serum. And so then the United States this was pioneered by the Mayo Clinic, although the original protocol was written by Johns Hopkins. So the Johns Hopkins investigators basically wrote a FDA IND protocol that allowed us to use convalescent serum all over America from patients that had survived SARS COVID-19 infection, that were 4 weeks out, and that serum could be collected via plasmapheresis and given to patients that were admitted to the hospital with COVID-19 disease. And this was done under this IND, this broad IND, which was given by the FDA. And the Mayo Clinic led this protocol, and they have given this convalescent plasma to, I think, over 6,000 people now. They have showed that it doesn't do harm, but real double-blind studies that really compare this convalescent plasma to non-immune convalescent plasma are still lacking. There are 5 or 6 randomized double-blind trials which have been initiated. I think 3 or 4 of them are in the United States. One is in the Bronx at Montefiore, one is here at Stony Brook, and there's a few other ones. Johns Hopkins also has one. Many of the trials didn't become operative until their individual institutions were through the surge. So these trials are now having problems recruiting. The trials were also basically hampered by the fact that the Mayo Clinic put out this compassionate use of convalescent plasma. Basically, they hired all blood banks across the nation to plasmapherese patients that had survived coronavirus. And so there was lots of convalescent plasma collected across the nation and given in a compassionate use protocol to patients. Also all over New York. Mount Sinai did it that way, and other institutions did it that way, because the emotional pressure on offering this treatment modality was huge. And so we basically, in an uncontrolled fashion, gave out convalescent plasma everywhere without ever knowing whether it works or not. And this is a good example of what press releases and social media can do. They interfere with evidence-based medicine. And it's my personal opinion that convalescent plasma is overrated and probably doesn't help as much as we think. And we would have that result if we, in an organized fashion, from the beginning on, would have only given out that treatment modality under double-blind placebo-controlled trials. But we didn't. We gave it out under compassionate use. So we do not have a clear answer as of now. The next thing that basically is very important is to develop vaccines. And this is a picture of the influenza vaccine. And this is to remind everybody that we have to make new influenza vaccine every year. We start in the winter based on strains that emerge in Asia. And then we basically grow up the strains. And we kill the strains with we grow up the strains in eggs. And this is what this shows. And doing that, you can grow up strains for millions and billions of doses. And basically, you recover the virus from the eggs. And then you formaldehyde treat the purified virus. And that makes the virus, then the virus is dead. And then you can inject that virus into humans, and that's your vaccine. And every year we do this, and every year we produce many doses that way, because the virus changes all the time. Now, you can do that with flu, because flu can grow in eggs. You cannot do this with corona, and you would also have a hard time making billions of doses that way. So what early on people have looked into the new vaccine platforms that have been developed over the last 15 years. The NIH has taken the lead in that, but BARDA has been very active in that, and companies have been very active in that. And the reason why people have developed new vaccine platforms is because everybody in anticipation of a pandemic always felt that we need to have a way of making a new vaccine fast. And when we are talking fast, we mean months. And now, more than ever in our entire history, these platforms are being used and are being put to task. And this basically shows you what these new platforms entail. And so the platform I'm talking about is these RNA-based vaccines. And the reason why these RNA-based vaccines are very lucrative is because you can make a safe vaccine very fast of a virus that's totally unknown. And so basically what you do is you generate, you can generate this RNA vaccine and basically very fast develop an RNA-based vaccine that you can scale up fast. You can do similar things with the DNA vaccine, but it becomes a lot more difficult if you want to make vaccines, if you want to make inactivated vaccines, or if you want to even make live attenuated vaccines or recombinant protein vaccines. These vaccines are difficult because, as you can see, the spike protein, for instance, is a very complex protein. So it's not that easy to make it recombinant and really mimic the virus and really generate a protein that induces those antibodies that you need. All these vaccines and viruses are based on inducing predominantly neutralizing antibodies, but are also hopeful to potentially initiate T-cell help, which will make the antibody response much more long-lasting. So protein vaccines are challenging because of the complexity of conformation, and these proteins are usually not simple proteins. Also, these proteins change all the time, and it's hard to scale up that production. Vector-based vaccines have been developed in recent years. The vector is usually an adenovirus vaccine, and some of the vaccines that are being used now are also using this platform. Inactivated vaccines, that's what we use for flu. These viruses require that you grow up enormous amounts of viruses, which sounds not challenging, but can be actually quite challenging and quite unsafe, especially if the virus aerosolizes and you could potentially have outbreaks just from growing this virus. And so these can be very challenging, and you need to identify ways of growing this inactivated virus, these viruses. That's either in cell lines or in flu that's in eggs. And the same is true with live attenuated vaccines. These viruses, these viral vaccines are usually the best vaccines. The measles vaccine is such a vaccine, but you need to be damn sure that that vaccine, which is still alive, is truly attenuated, so does not flip back like it happened with the poliovirus, so that it stays attenuated. And it takes time to make these kind of live attenuated virus vaccines. They are working on that. There's a local company that was spearheaded by Eckart Wimmer that are making a synthetic virus that is hopefully going to be live attenuated, and they're working on that. These viruses can be very successful, very good virus vaccines, can give you long-lasting immunity, but they are harder to make. And so right now the new platform is these RNA-based vaccines, and I'll talk a little bit about that. So the Moderna vaccine, the vaccine which is spearheaded by Moderna and the NIAID, that's a messenger RNA-based vaccine. Then you've probably heard that the Germans are making one together with BioNTech and Pfizer, and that's also a messenger RNA-based vaccine. There is a Chinese vaccine that's out there. And then there is the Oxford vaccine. This Oxford vaccine is an adeno-based based vaccine. This virus is already in phase 3. I believe it's being tested in Brazil. This virus is in phase 3, is also being tested now. And this virus, I believe, is also being tested in South America, is also in phase 3. So these vaccines are moving forward. You want to have as many vaccines as possible. There's also a Russian vaccine out there. Nobody knows where that is standing. There's Chinese vaccines that are in phase 3. And I don't think that there is any problem with making as many vaccines as possible, because we will need billions of doses. And so it's in our interest to have not only competition, but to also have a very variable portfolio of vaccines that are based on different platforms. So the RNA vaccines are basically, you generate these synthetic RNAs, you package them in something that allows this RNA to be taken up, and then this synthetic viral RNA, which in this case would probably be the spike protein, is then basically taken up by the host cell. The host cell expresses this protein and then expresses the protein on the surface, and then makes both a T-cell response and an antibody response. There is some concerns with instability, and there's some concerns with low immunogenicity. These viral these RNA vaccine platforms have been tested in some preliminary human vaccines and have been tested in animals, but there's not a commercially available vaccine out there yet. The reason why people like these vaccines is because you can scale up fast. You never have to produce the whole virus, you just produce this RNA. And it's pretty safe to do this, and it's easy to make a lot of doses. And you can do tricks where you have self-amplifying messenger RNAs through molecular engineering. That means that once the RNA is taken up, it actually self-amplifies within the cell, so you can get higher load of the RNAs and hopefully a better sort of efficacy. And as I said, fast scale up is possible, and cell and egg free, and so it's easier. You don't need these intermediate growth platforms, which is very important. And the other thing is that it can induce D and B cell immune responses. It's not clear the impact on the innate immune response, so it's not clear whether you could also induce an innate immune response that's going to clear all this messenger RNA through PAMP recognition, and that would basically render the vaccine non-efficacious, and that's one concern. What's the biggest fear about vaccines? Well, the biggest fear is probably that the social media will be used to spread conspiracy theories by dogmatic anti-vaccine opponents. But I think with the recent scare of COVID-19, that the majority of the Americans and the majority of people worldwide will actually get vaccinated. Because we all have to understand, a vaccine doesn't do anything. It doesn't protect us. What protects us is vaccination. So unless we can convince people to get vaccinated, the vaccine doesn't help us. So we would hope that that these conspiracy theories, which for sure will be spread by dogmatic anti-vaccine opponents, will be shouted down by a pragmatic, rational majority of people who will understand that vaccines, even though they are never riskless, the overwhelming data is that they protect and don't hurt us. There is always some concern about disease-enhancing antibodies when you have a vaccine out there. As I explained to you, this is a concern in dengue, and this has hampered vaccine development in dengue in order to curtail those pandemics, which basically happen every year worldwide. So basically in dengue, if you make antibodies against one dengue type and you get infected with another dengue type, then the antibody from that is pre-existing can actually enhance the second dengue infection and make it worse. And so there are concerns that when you vaccinate people that potentially have already seen dengue, that you could make it worse. And so you need to be very careful and monitor very careful for disease-enhancing antibodies. There's some data that disease-enhancing antibodies can occur in coronavirus infection. It doesn't look like in COVID-19 we saw a lot of that. My biggest concern is the real risk of that the vaccines will not work. So many vaccines have an efficacy between 40 and 60%, like the flu vaccine has an efficacy between 40 and 60%. And that's mainly because there is a significant percentage of patients that are old, have comorbidities, that don't make good antibodies. And they don't make neutralizing antibodies, so they don't contribute to herd immunity. But as long as many people get vaccinated, that doesn't matter, because all the healthy people will make antibodies, will generate herd immunity, and will protect the people that don't make antibodies. So that's one way of not making, having a vaccine not work. The other one is that sometimes a vaccine makes antibodies, so elicits neutralizing antibodies, but the neutralizing antibodies don't really protect. And that is something you don't really know until you test this in a pandemic situation. And that's why these phase 3 trials are going to be very, very important to really establish whether vaccination protects people. That means if you have a pre-existing immune response, is that immune response going to be efficacious and fast enough to prevent the devastating effect of COVID-19 and abort the infection early? overwhelming data from animals is it will, but we don't know until we test the vaccine. And so that's why we need to go broad. We need to not rely only on one vaccine. We need to test different forms of vaccines. We need to do good clinical trials, not go through shortcuts, but really, really go out there and generate as many vaccines as possible and do good clinical research in order to establish which vaccine is the best. And this is the data on immune-enhancing antibodies. This is data that was published early on. And the concern is that antibodies against the spike protein could potentially, if they are not in high enough a dose, help the virus to get into a cell by promoting FC-related uptake of the virus. So this is one way how you can produce disease-enhancing antibodies with vaccines. And we need to look out for this and make sure that we don't do this. We also, of course, want to make sure that we don't make an antibody that cross-reacts with something and causes damage, so kind of an autoimmune disease, but we don't have any evidence about this from current COVID-19 disease. Okay, so the last of the talk, I want to spend a little bit talking about the epidemiology of pandemics and comparing COVID-19 to other pandemics. So globally, we see emerging and re-emerging infectious diseases all the time, and we see pandemics all the time. And as you can see from here, the majority of pandemics that we see are caused by viruses and not by other pathogens. Although we do see pandemics also caused by fungi. For instance, Candida auris has caused a pandemic worldwide. It's popping up in different places. We have seen it caused by bacteria. We have pandemics with multi-drug resistant bacteria popping up everywhere, but we see it more and higher case numbers with viruses. So this is the death toll of pandemics in the human history, and the biggest death toll is, of course, the Black Death, which was caused by bubonic plague, and basically decimated the European population, the worldwide, but especially in Europe, the population. It led to, in some places, 30 to 40% of the population died. This is how big the plague was. And it basically lasted a good 4 years. And so it didn't go on forever. And this is classic of a pandemic. Pandemics are usually not smoldering. They surge, they happen, and eventually human immunity deals with it, and some die and some survive. Smallpox was also a big pandemic, and smallpox came in bouts in Europe, and smallpox and the trauma of smallpox is responsible for all of the vaccine endeavors and all of the research, but all of the early vaccine findings by Jenner. But we all have to remember that the Chinese vaccinated against smallpox many thousands of years before us, because they knew that with variolation, with scratching, with small doses under the skin, you could protect people from smallpox. Smallpox also had a huge impact on Europe, and because this is a talk for female scientists, I want to highlight that big vaccine proponents of against smallpox were the three famous European emperors, that is Katharina the Great, Maria Theresa of Austria, and Queen Victoria, all three of them were big proponents of vaccination and pushed it, and they all got vaccinated themselves. Then the Spanish flu, we all know about that. And I'll talk a little bit about that because I think we can learn a lot from flu pandemics when we talk about COVID-19. Now, plague came back several times, not only in the Middle Ages, but also earlier. We all know about the big pandemic of HIV, which is still going on. So it's one of those pandemics that took a long time. It spread very differently than the other pandemics, and that's part of the reason. But we learned a lot from dealing with this pandemic, and we learned about getting organized, we learned about feasibility, and we learned about setting milestones. And then there are all these smaller outbreaks that led to a lot less death. So the Spanish flu pandemic caused about 500 million deaths, and 1/3 of the world's population became infected. At least 50 million people died worldwide, and in the US, 670,000 people died in the US. The mortality was high in young kids under 5, and was high also in 65-year-olds. But it was also high in the 20 to 40-year-old, which was a very unique feature of that pandemic. And whenever we deal with a pandemic, we always want to know not only how many people die, but who dies, because that's actually very important. And it's important in 2 ways. It's important in predicting how it will spread, and it's also important in predicting the ultimate damage to society. One thing that we learned from the flu is we learned that it came in waves. And this is important, and this is why people talk about waves here. And this shows you that when you deal with a pandemic and you react to the pandemic, unless you keep whatever you are doing up, the minute you start loosening any kind of regulation, the pandemic can often resurge again. And this is what we're seeing now. For those of you who don't know what Spanish flu is, I want to remind you in Downton Abbey, Lavinia dies. She's the first wife of the heartthrob, Matthew. She dies of Spanish flu. Okay, so in the 20th century, we had more than one flu pandemic. Spanish flu was the first one in 19 and caused the most death. 33% of the world population got infected. That just shows you that we had a much smaller world population at that time. And the death worldwide, there's some debate, but anywhere between 20 and 100 million. The case fatality rate, and it's important to understand case fatality is means case is the patients who are symptomatic. They was about 2.5%. That doesn't mean that 2.5% of the people died, but 2.5% of the people that got sick died. We had smaller outbreaks involving much less people, but crossing continents. There was an Asian flu, a Hong Kong flu, and then we had the Mexican flu. That was the H1N1 outbreak. I remember that very, very clearly. I was already an infectious disease consultant and very active. And I remember how that evolved in New York. And we have learned a lot from that outbreak. Many people got infected. Also, 500, many more people got infected than that. In New York, 500,000 got infected, but only 8,000 died. Across the US, it ravaged between August 2009 and March 2010. I don't know whether you remember, but even the bodyguard of President Obama got infected because they went to Mexico, where it started. In New York, in New York City, we had 41,000 hospitalizations and 2,000 deaths. The 8,000 deaths, that was in America. So one thing to understand about influenza is the reason why we see pandemics with influenza is because influenza is a virus that has 8 segmented RNA genomes. And that's the problem, because this segmented genome loves to rearrange. And when it rearranges, then you get an antigen shift. So an antigenic drift is when you get mutations in the critical surface molecules, but an antigenic shift is when you get a real rearrangement of these segmented genomes. And that happens when a host becomes infected with 2 different flu viruses. So there are many different flu viruses, and we label them based on the 2 most critical surface molecules. One is the heme agglutinin, and the other one is the neuroaminidase. One is important for binding to the receptor, and the other one is important to being released from the cell. So they're very critical surface molecules. And they mutate all the time because they're on the surface, and we make antibody response to them. So there's enormous selection pressure on them. And depending how they mutate, they generate different mutants and different classes. And we name them, and I think there are 19 different H molecules, and I don't know how many different N molecules, I forget, but there are many different ones, and those are what these numbers are. And so different flu strains have different variants of these surface molecules. And the surface molecules variants also determine the host tropism, because you bind to the receptor structure in order to get into the cell. So that means that the flu virus that infects the dog usually doesn't have a high affinity to your receptor. Or the flu vaccine, the flu virus that infects the birds doesn't have a high affinity to your receptor either. It binds to a completely different receptor. But every once in a while, somebody is unlucky and gets infected with 2 different flu viruses at the same time. And one flu virus could be from a pig or from a bird or from a dog, who knows? And then once they are together in the same host, they can rearrange and they can generate a new virus. And that new virus becomes pandemic very fast because it has a full new set of H and N, never seen by human immune systems before, no pre-existing immune response. And if that virus is highly transmissible, if that virus is virulent, then you have a problem. And we always knew that, and we always watch out for that. Every year we go into Asia and we look at all the different viruses, all the flu viruses, and we make sure that we're not dealing with a new flu virus. And based on the flu virus that emerges in Asia, we make our new virus, our new flu vaccine. Now, this only happens with influenza A. There's influenza A, B, and C. And your vaccine has A strains and B strains. The B strains are just as bad as the A strains, but the B strains only infect humans and seals. So you do not see antigenic shift because there's just not enough jumping between species. But the A viruses, the A strains, they infect all kinds of different hosts. They infect dogs, cats, horses, birds, anything. And so chicken, so the chance that you can get a shift is much higher. And that's why all the pandemics were A strains and never B strains. Okay, so now looking at this, we're going to ask, now can that happen with coronavirus? Now, I already told you before, the coronavirus is also a virus that jumps between animals and humans. And it infects bats, and it infects all kinds of animals. And most of these animals, if you give me the name, I have to Google them to see how they look. But it's usually raccoons and civets and all kinds of animals. And in Asian markets, some of these animals are sold and they're brought there alive and slaughtered there alive. And there's a lot of people there. And that's how these infections emerge in these wet markets. And that's also what happened with SARS, most likely. And this is also what happened with COVID-19. The coronavirus does not have a segmented genome. So we wouldn't see the same kind of rearrangement and reemergence every year, but there is description that this coronavirus also have an ability to recombine, both homologous and non-homologous. So if the virus is going to jump back and forth between animals and humans, we can also anticipate, especially once we start vaccinating, that we could see reemergence of new strains. So we need to be aware of that, and we would have to monitor this very carefully. And that's the reason why Fauci said he doesn't think we will ever be able to eradicate this virus. And he is right. A virus that infects a whole host of different animals is very, very difficult to get rid of. You can't get rid of rabies. You can't get rid of flu. You can't get rid of corona. You can get rid of measles. Only infects humans. Every human is vaccinated against measles. Measles is eradicated. You can get rid of polio. Infects humans. You can get rid of smallpox. We did. Why? Because it only infects humans, and we vaccinated successfully. So we do not see smallpox cases anymore. So, but this is not going to happen with corona. So pandemics are not new to New York. Before this COVID-19 outbreak, we had a measles outbreak in 2018 because people stopped vaccinating, and we had measles outbreaks in the Orthodox community who didn't believe in measles vaccination. We had Candida auris outbreak, that's a fungus. We had the Zika virus scare that emerged in South America, and we were very scared about that because that's a mosquito spread virus. We had the Ebola virus. We were also scared about that because it was killing so many people in Africa, and all of a sudden we had 3 patients in the United States. But then we realized, and the good thing about the Ebola virus outbreak in the United States is that it trained us to put everything in place and to rehearse how we deal with a highly infectious virus. But it never came to an epidemic. We never saw a lot of cases. All hospitals got prepared. But we contained the virus in Africa, and Ebola never became a pandemic. We had measles outbreaks before, and then we had the pandemic flu outbreak, H1N1, in 2009. It started in Mexico. This outbreak originated from a new strain, which was a rearranged strain between a pig flu strain and a human flu strain, and generated a new strain that hadn't been there before. But there was cross-reactive immunity to an older strain that had occurred in the 50s. And that's why this pandemic never led to a lot of deaths, because the only people affected were kids that didn't have pre-existing immunity. All the old people, which are usually the ones that die of flu, had pre-existing immunity. And that's the outbreak, spread very quickly. I remember the weekend it was announced, and they were thinking about closing down the subway. It was under Bloomberg, and then they made the decision that it wasn't worth closing down the subway because it was already out. It had appeared in schools in the week, and then there were soccer matches all over the tri-state area, and within days, it was everywhere.
And so the one thing we can learn from that is how quickly upper respiratory virus can spread. There were approximately 60 million cases over the course of the following year. So it really spread all through the United States. It resulted in 275,000 hospitalizations, but not more than 12,000 deaths, which is low on the flu side. And the reason is because of this pre-existing immunity. Now, in New York, we were the first one again, hit heavily. We had 8,000 cases. We were not prepared. We weren't vaccinated in the hospital. Yeah, it was recommended to get vaccinated against flu, but most healthcare providers thought that they were invincible and didn't get vaccinated. So we had a lot of healthcare providers that got sick. It was actually terrible because we had whole hospitals that were running into problems because their house staff was getting sick. We saw over 41,000 hospitalizations and 2,000 deaths. Nothing compared to what we saw in COVID-19. But it trained us. It trained us to get organized. And we very fast developed a vaccine. The outbreak started in March, and we started vaccinating in October. And we vaccinated 195,000 kids in New York, in 1,200 schools, and an additional 50,000 people at other locations across the city. More than 700 schools had to be closed during the H1N1 outbreak. It stuck us in April and May, and we vaccinated in October. So this is just to tell you about the timeline. But we didn't have to invent a vaccine. We just had to make a new one, but we knew exactly how to make a flu vaccine. The vaccine was rolled out in October, yeah. Okay, now the Zika scare in the US was a big scare because that happened during the time of the 2016 Olympics and in Brazil, and that's where it started. What makes this Zika pandemic different is that it's a mosquito spread pandemic. And the scary thing about the Zika pandemic was that it infected unborn babies. So women that were pregnant gave it to their children, and the children ended up with horrible malformations. And the reason why America got very scared is because it was spreading through mosquitoes, and those mosquitoes were also present in the United States. And there was a concern that we would find a mosquito jump, so it would also infect our local species, and that we would see Zika infections across the United States. And so there was a lot of back and forth between whether we should go to the Olympics or not. I don't know whether you remember, but all people got out of their cruises. Nobody wanted to travel to the Caribbean because of Zika. Pregnant women canceled their honeymoons. And there was a lot of surveillance instituted in the United States early on to make sure that we did not have an infected mosquito population. And they very aggressively sprayed mosquitoes. And it never came in big numbers to the United States. So now we're going to talk about COVID-19. This one started in 2019 in December, and I want to sort of show you the time frame when it started. So it started in China sort of in the beginning of December, and they were having single cases appearing in hospitals in Wuhan, and all of these cases they figured out were connected to this seafood wholesale market. And it took them some time to figure out that it was connected to this market. Now you can say that the Chinese were sitting on this and that they were not reporting correctly, and that the CDC in China, which is the NHC, wasn't properly documenting what was really going on. And some of this is probably true. But I want to remind everybody, if this would have started in New York, and if we would have had upper respiratory infections of unknown origin, of unknown pathogenesis appear in different hospitals across New York State, it would have also taken us some time until somebody would have said, darn it, what's going on here? We have young patients that are dying of an infection that we can't diagnose. So it's not like we within days would have made this diagnosis either. When this usually happens, there's a procedure in place, and it happens that the ERs then report upper respiratory infection of unknown origin. And then in New York, the New York Department of Health would step in and would try to identify the pathogen. And then they would put out an alert to all the other ERs and then basically establish whether we have a new virus out there. Understanding that you have a new virus, informing the CDC you have a new virus, the CDC informing the president that you have a new virus, and the president announcing on national television we have an outbreak. That is a whole chain. And how that would have played out here, we don't know either. What we know is that the China, that by December 31st, they had a case definition. They announced an outbreak at the beginning of January, but they didn't think that the outbreak, that they had major human-to-human transmission. They thought that, well, that this was all related to that food market. And then it took them some time until they really sort of said, okay, we have human-to-human spread. Because if you have an outbreak that can all be led back, if everybody gets infected by the same animal, then you just have to get rid of the animal. Once the humans give it to each other, then you have the potential of a pandemic. And I think this is where China and in part the WHO screwed up. They were downplaying the spread, and they put their physicians in prison that said that there was a lot more cases. And so this is where I think they can be faulted of not being open enough. But they did release the coronavirus sequence very early on, so they didn't hold that back. Then, January 14th, we started seeing outbreaks outside. So this is the one, December 31st, WHO is informed of cases. January 11th, they have the first death secondary to this coronavirus. So it even took China quite some time until they really saw death. And then January 30th, WHO declares a public health emergency of international concern. And basically, we shut down all travel to China. And in February 7th, very tragically, the Chinese doctor who tried to warn about the virus and was put in prison instead died of the virus. And February 29th, the US reports its first COVID-19 death. It takes until March 11th that the WHO declares a pandemic. Now, this is not so unusual. The WHO always lags behind. I think the real problem is that the CDC in January 30th only shut down travel to China, and somewhere in February should have shut down travel to Europe and didn't do that. And we could have been spared a lot of misery if we would have curtailed that much earlier. So this is how it evolved in New York. New York, early on in its recognition, reacted. So January 17th, the first case appears in Washington State. January 26th, New York Department of Health launched a website. February 2nd, New York Department of Health started a hotline. February 29th, Wadsworth approved the first FDA test. On March 1st, we see the first case in New York, which was a woman that traveled from Iran. And within days, we see cases appearing in New Rochelle that we immediately cannot link to travel anymore. And that's where the problem starts, because then you know you have community-wide spread. We saw the first case March 7th. And as you can see, as it evolves, March 10th, we started containment in New Rochelle, then we shut down Broadway. March 16th, we started closing all schools, and New York State basically went on pause March 20th, and we started canceling elective surgery March 22nd, and March 23rd, we were basically demanded to increase our bed capacity by 50%. So it evolved really, really fast. And I think the thing to understand here is that a lot of this was based on a fairly low number in the hospital. So these are the numbers in the hospital. So March, but they then rapidly increased. So this is where we are, March 7th, when we have our first case. This is where we are when we start shutting down the entire state. So by that time, we had that many cases. And you could see that the number of new cases were like doubling very fast. And then we basically went into the surge, and we peaked in the surge probably at the beginning of April. I remember I was on call, and by that time, we basically had instituted 6 consultation services. And these are our numbers. In peak times, we had 562 patients hospitalized, and we had like 128 patients on ventilators. So we had 423 patients hospitalized in peak times, and we had 128 patients on ventilators. All over Suffolk County always lagged a few days behind New York City. So about 6 days. New York had their first case March 1st. We had our first case March 7th. So we were about a week behind them, which gave us a little bit more time to prepare, but we were hit just as badly. And we ended up also having 43,000 cases and lost nearly 2,000 patients. So this is just sort of now a brief review of how you have to stop a pandemic. And I think there's a lot of lessons to learn from this. And STOP stands for you need a strategy, which is based on what you know now and learn along the way. And that's very important because a pandemic is incredibly dynamic. You need a team, and that has to be multidisciplinary. You need organization, because anything you do has to be organized to implement it. And you need to understand, and you need to predict the barriers, because it's very difficult to implement a lot of these things. And then you need a plan that is based on data, guidelines, and regulations. And some of the guidelines and the regulations ended up being wrong. And that's why there's so much doubt now when we come up with new plans. And I think in the future, we have to get better at communicating the confidence that we have in the plan that we basically put out there. So one thing that's really important to understand when you deal with a viral pandemic or with any pandemic is you need to understand the transmission. And so any virus that's airborne with small droplets, that is probably the worst pandemic to deal with. And the reason is because it flies far and it's much harder to contain. So the first mistake that was made by the CDC and by the WHO is that it took them very long to understand that we were not dealing with a large droplet airborne, but we were dealing more with a small droplet airborne. So that it was more contagious than normal flu. And although we kind of sensed it, it wasn't really spelled out. And this is actually very important to spell out because it has big impact on how you're going to try to prevent it. So these are other ways of transmission. And as you can see, once you have sexual transmission or blood, like HIV, you know, the dynamics are very, very different. Fecal oral can also spread very fast. And that's what happens with Ebola. But it's also, in a way, easier to contain because you can just keep the people at home. And the other big route of transmission are insects. And there are a lot of pandemics that are spread by insects. Dengue, Zika, West Nile were all spread by insects. The bad thing is that insects, you can't vaccinate insects. But the good thing is that with modern insecticides, you can often curtail the insects. And you can spray and you can make sure that they can't breed anywhere, and that can curtail the transmission. And the reason why Zika was such a difficult pandemic to control is because it was spread by an insect that was biting during the day and during the night. If the insect only bites us by night, which is the malaria insect, then it's actually very easy because then you just tell people to sleep in under a mosquito net. But if you have mosquitoes that bite 24 hours a day, then it's a little bit harder. So you need epidemiologic data in order to sort of judge a pandemic. And the epidemiological data that came out of China was that the median incubation time, so the time from when you get infected until when you're symptomatic, was 5 days. And that the mean time from illness to when you appeared in the hospital was 3 to 4 days. And that the mean time of onset to death was 14 days. And based on this data, we recommend that the length of quarantine should be at least 14 days. That's what this is based on, and we still stick to that because we think that within 14 days, most of the patients that got sick will actually be sick. And that once they're over 14 days, they are probably not spreading the virus anymore. Now, the other big label that is tossed around is the efficiency of transmission, and that's the R0 value. And this basically tells you how many people, on average, become infected. So it tells you how infectious your virus is. Now, to give you an idea, the most infectious virus alive now is probably measles, and the R0 of measles is 18. So it's very high. Initially, they were talking that the R0 was 2.2 for this virus. So people got really relaxed and said, okay, per person infected, they're going to infect 2 people. That's fairly easy to contain. But it turns out that the initial calculations were not correct. And when they recalculated based on better data, they actually came up that the R0 was higher, and that was more like around 5.7, which also explained why it spread so fast. So from an epidemiological point of view, we need to get this under 1 in order to really stop the pandemic. And so that's the number that we use to measure to see whether anything that we do is helpful. Now, the serological response, meaning your antibody response, doesn't really help you to diagnose the patient, but it helps you a lot to determine the number of infected patients that are not symptomatic. And you need to know that number in order to come up with better calculations and in order to really judge the risk. So this is the data that I was talking about. So initially we're relying on the epidemiological calculations of the Chinese, but then they went back and recalculated based on patients that left Wuhan and went to other provinces in China, and basically started the pandemic in those provinces. So these were patients that left Wuhan infected, went to provinces that didn't have any cases yet. And so based on this math modeling, they could much better calculate how rapidly it was actually spreading. And based on that, they found that it was spreading much faster and that the real R0 was 5.7. And this is important for early effective surveillance, contact tracing, and quarantine, because you need to know how many patients are asymptomatic, and you need to know how much of the transmission is driven by patients that have the disease and how much is driven by patients that weren't symptomatic. So by using these new calculations, they knew how many people had flown to that area, and how many people of those that had flown to the area ended up getting sick, and how many ended up not getting sick. So they could get a much better handle on also understanding how many asymptomatic people were spreading. So asymptomatic people are people who have the disease, don't know they have the disease, but still infect others. And what is very clear is when 20% of transmission is driven by unidentified infected people, high levels of social distancing efforts will be needed to contain the virus. And this is what's happening now in the southern states of of the United States. We have a large number of infected people who don't know that they are infected, and they continue to transmit the virus. And if you don't know who is infected, then quarantining and tracking alone doesn't help. So the minute you have lots of patients come to the hospital that don't know where they got it from, it becomes almost impossible to contain the infection, the pandemic, with contact tracing. Then you only have one option, which is extreme social distancing. That means based on data, based on calculating how many patients you have, how many people you have per 1,000 or per 100,000, you calculate how big a crowd size you allow. And that's what's happening to us. So here you can see this is 50% undetected, this is 20% undetected, and this is 0% undetected. And here you can see how difficult it is to reduce the overall rates of new contacts, of new infections, if this number is very, very high. So even if you quarantine everybody, you are still going to have huge numbers of infections because you are only quarantining the people that you know. So if you don't identify everybody who's infected, which is happening in the United States because we don't have testing capacity and we don't have a system in place to really screen everybody, and if you then have lots of people running around who don't know that they have the infection, you get a pandemic that you can't contain. And that's why it's so important to comply with quarantine and to comply with social distancing rules. And that's why masks are very important. So we knew early on that we had patients that were not symptomatic because early on, the Japanese government flew out 565 Japanese citizens who were in Wuhan. They flew out everybody. And they tested all these people. And then they checked who of the ones that were positive ended up becoming infected. And they found that 33% of the ones that were positive that never developed symptoms. So they knew based on that data that there were people running around COVID-19 infected, but not symptomatic. And this data was confirmed by analysis from people from the cruise ship, because on the cruise ship, they also tested everybody. In the cruise ship, it was less, it was 18%, because the majority of the population on the cruise ship was old. And being asymptomatic is a lower chance if you're older. Now the numbers are 40% because we have widespread infection also in younger people. And younger people are much more likely to be not infected, to be not symptomatic. So the other thing that we can learn from China is that we can't do what they can do, because China is not a democracy, and they're proving this every day. So China put basically 1.4 million people on house arrest. They basically opened 50,000 hospital beds within 2 weeks, and they redeployed 40,000 healthcare workers. And we are not able to do any of that. We can't even organize ventilators across the nation. We opened 50,000 hospital beds in New York. We did that. But we could not redeploy healthcare workers easily. We had 90,000 volunteers sign up to help in hospitals, but we just didn't have a system in place to actually get them into the hospital. We also couldn't really use the National Guard the way we should have been able to use. They built this huge Javits complex, but the Javits complex was never used the way, was never very effective because it didn't have oxygen and didn't have x-rays. So it would have been probably much better if the Army would have just helped us in the hospital, if they would have just doubled us up in the hospital. But we didn't have that mechanism in place. What we had in place is a hospital incidence command structure. And this is something that a lot of people don't know about, but this is something that is demanded by George Washington Bush. It was put in place in 2003, and he created this National Incident Management System. And within this National Incident Management System, that provides a framework for interoperativity and compatibility to do a flexible, basically, hospital incident command structure. And what this basically is, it's an administrative structure that is pre-existing, that is activated in the setting of an emergency. And this hospital incident command structure is present in every hospital in America. And that saved at least us, and that saved New York. I mean, we still lost a lot of people, but this allowed us to become organized very rapidly in a very, very systematic way. And so our commander was Mark Sands, who is the CMO of the hospital. And as you can see, there's a pre-existing infrastructure in place. And when you activate this incidence emergency structure, you just fill all the spots. And people are trained to step up to this leadership role, and in a very organized way, you run the entire hospital operation under this command structure. And that's what we did. And part of that command structure was there were logistic people in there that basically made sure we had supplies, that made sure we got money from the state, that made sure we had information technology in place. There were finance people in place. There was operational people in place. For each medical branch, the surgeon had somebody, the OBGYN had somebody, internal medicine had somebody. I was somewhere in there organizing the trials. There were people in there from nursing, organizing nursing. There were people from labor in there. The press was in there, the dean. All in all, we were about 100 people on the phone call every day at a sharp time we started. And every day in a very systematic fashion, we organized the day of the hospital. We anticipated the problem ahead of us and dealt with whatever was happening. And this way we're able to basically maneuver, manage the whole operation through the pandemic and deal with all the supply shortages, which of course hit us across America, and anticipate what we needed, anticipated how we get organized. And this was, I think, what really was done extremely well at Stony Brook, and also very well at all other hospitals. And the hospitals communicated with the state and also communicated with each other. And he was the master of communication, and he basically showed New York that the most important thing in a pandemic is that you have to talk to people and put all the data that you have out there. And I think it's very important to understand that you make mistakes in a pandemic, and that these mistakes need to be corrected along. So these are some of the things we did. We immediately started the field ER. So we took all the triaging outside of the hospital, put it in tents. Everybody in there had to wear masks, had to wear N95s just to make sure that people weren't getting infected. This shows you data how the government sort of left us hanging, and how our government was way behind other governments. Like March 19th, we only had tested 100,000 people, whereas Germany at that time had already facilitated to test 300,000 people, which was 3 times as much as we did. So it was, it shows you that a lot of things that we did right on a local level were done wrong on a national level, because we were just incapable on a national level to deal with all of the testing issues. We were just not able to test the number of patients that we needed to test. And this shows you also that America was vulnerable, because we drive a hospital system with very few beds in order to be financially efficient. And so we only have about 2.4 hospital beds per 1,000 inhabitants, whereas Germany and South Korea have 3 times as many hospital beds. And because of that, and that's something that Como understood very early, we had to immediately double our hospital beds in order to meet the criteria, to meet the demand. So what have we learned from the H1N1 outbreak? From that outbreak in 2009, we learned that we could not contain it. It was respiratory, and it went so fast that it was impossible to contain. We learned that healthcare providers became infected and gave it to their patients. So we knew that healthcare providers had to be really protected. And we knew that some of the healthcare providers, some hospitals had so many healthcare providers that got sick that the hospital got into trouble. So we knew that this was very important. We knew that the death rate was lower than anticipated in this, so that saved this. And we knew that mass vaccination efforts deployed within 6 months. So we were able to get organized very fast. Now, what have we learned so far from the SARS outbreak? We also learned that we could not contain it, that it went so fast and so rapid, it was impossible to contain. We learned, this is just a couple. We learned that we protected our healthcare providers. Sorry, this is incorrect. We actually protected our healthcare providers. We had 400 healthcare providers that got infected during our epidemic. None of them, as far as I know, died. And the majority of them got infected, most likely in their communities. So we did have shortage of healthcare providers. We had to fly in nurses from Syracuse, but that was because we doubled our beds and we had so many intensive care unit need that we didn't have enough nurses. The death rate was high, and we still don't have mass vaccination. I think what we really have learned is that if we want to make America great again, we need to change the way we do things. And I think having been in the pandemic, one of the things that has been lacking all along is we need a national strategy, because we, on a local level, can only do so much. But, you know, even though New York has now everything controlled, we all see that if the other states are not controlled, we can't open up our universities properly because all of the people are coming in from other states. So we need to all give up on this federal identity for some time and really come together as a nation in order to fight this pandemic, because it's not individual states, it's the entire nation that's affected by it. And we need to become a nation that gets in line and follows instructions. And it starts with leadership modeling and modeling guidelines, role modeling guidelines. Nobody should get on television without wearing a mask. And we need to understand that social disparity and unrest will hold us back. People going out, demonstrating, people, you know, not following instructions, people not having access to healthcare, all of these things, they now backfire and they need to be addressed. So I think we're a long way from having this under control. Now, to end on a good note, evolution has given us the tool to beat this virus with our immune system. Most of us are going to do all right. And if we had nothing, if we had no regulation and we would just let it happen, then we would probably lose between 1 and 2 million Americans, and the rest would survive. And this is how pandemics have basically happened in the old times. A significant percentage of people died, and the other ones came out of it and were stronger. And we are not willing to accept this, and we can probably not afford it either. And so that's why I think we need to all get together and fight this. And that's it.
Thank you very much. That was an outstanding lecture, and I've gotten several comments in the chat from people saying that they really enjoyed that. It was very informative. So thank you very much. I have a couple of questions that have come in.
Sure.
And I'll ask those. The first one was, can you comment on any post-traumatic effects of the COVID-19?
So physiologic and emotional. So physiologic, so the bad thing about this disease, and I didn't emphasize that enough, people focus very much on the death rate. And you know, you can take sort of play the devil's advocate and can say, well, 160,000, that's big, but it's not sort of outrageous. And it's not. I mean, we lose 600,000 people to heart attacks every year, and we don't seem to lose sleep over it. The problem is the morbidity of this disease. So the problem is we lose a lot of people to flu, but it doesn't lock down the healthcare system. You know, when we have flu, we don't have people going to church and infecting the entire church. And with COVID-19, we have a serious proportion of people get really sick. And get really sick means they come to the hospital and they need to be either intubated or need to be on high-dose oxygen. They need to be in the ICU. And that number basically is what the hospitals are really struggling dealing with, because these patients absorb, you know, a lot of healthcare energy, and they end up getting a lot of sequelae. They get strokes, they become hypercoagulable. A lot of patients, not a lot, but a significant percentage of patients have permanent lung damage afterwards. That means they never feel that they have the same vital capacity and their breathing ability afterwards. So there's a lot of damage that stays after the patient leaves the hospital, and we don't have a full understanding yet how big that damage is. But it's for sure much bigger than flu. We have flu outbreaks all the time. And, you know, the people that get affected and die, they die, and it's sad, but it's contained. It doesn't affect the whole community. That's not the case with COVID-19. With respect to the emotional damage, I think the emotional damage was huge, and because we were so in the midst of this incredible pandemic, we probably spent the least time getting organized to deal with that. And so on the patient side and on the family side, we were aware how hard it was for the patients and for the family not to be able to visit. And this did not only affect the patient with COVID-19, but I know lots of families that lost loved ones who were probably already sick before during that time and were not able to be there with them. I know people who were diagnosed with cancer and had to go through this all by themselves because hospitals had these very strict policies that nobody could come along. You know, you couldn't have a baby the way you used to have a baby. You know, all of these things created the sense that things were different. Now, some of this damage, not such a big deal. Some of this damage is huge. And it was really hard on elderly people to be in nursing homes for months not being visited. Mm-hmm. And so I think that part of the emotional damage is in part still going on because there's still a lot of people that suffer tremendously from the social distancing regulations. There's a lot of domestic abuse. There is disabled, autistic kids not getting the care they need. There's elderly people not getting the care they need. There are people falling into depression. So there's a lot of damage. On the healthcare side, I do think that the nurses in the forefront that had to deal with, especially in those 3-4 weeks with surge, and the physicians, you know, to deal with losing so many patients, dealing with so much angst, anxiety on the patient's family side. If I would do this again, then I would demand that anybody taking care of patients has to get on an anonymous call once a week just to talk about it. Because I think that damage, we don't know yet how affected they were. But, you know, healthcare providers were really scared to get it themselves and give it to others. But I never saw anybody walk off the job.
Wonderful. We have one more question for you, and that was, how about, we're talking about the cost of the vaccine when it becomes available, and what will people who don't have some kind of health insurance or some way to pay for it, how will they be able to get the vaccine?
Oh yeah, I'm sure that, I mean, all the testing is given out free. I mean, we spend trillions on our economy. I think it's going to be, you know, peanuts compared to what we're losing in the economy buying the vaccine for non-insured people. I don't think that that's going to be an issue. The question is, you know, how much vaccine are we going to have available when, and how are we going to employ it? There's going to be legal questions about, are we going to mandate vaccination? That's going to affect us in the hospital because we tried mandating flu vaccination after 2009, and the nurses union blocked us. And so that's why Cuomo put in place that you have to wear masks in flu season. I don't know how that's going to work. Whether, you know, health insurances are going to demand that you protect yourself, or going to give you a bonus if you get vaccinated. I don't know. I think legally there is probably the possibility of mandating it from certain groups. Private hospitals for sure can mandate it of their employers. They put it in the contract. State hospital can't do it. my feeling is and that's also why I think they didn't go for, there was some discussion whether they would do the phase 3 vaccine trials just with a small group of people that get voluntarily infected. And there were actually 30,000 people volunteering for that. And I think that's part of the reason why they didn't do it. They wanted to really try the vaccine in the natural situation. And really see whether it can curtail the curve. Also to have an argument then, because as I said, vaccines don't do anything. Vaccination protects. So we need 99% buy-in. We need to basically shout out and be louder and more effective than all the conspiracy people. We need to really, I think anybody who's for vaccination has to like step up and make this known so that that we will be able to get the majority vaccinated.
Okay. One last question, and this is a bit of an issue coming up very shortly, and that's about schools. A lot of people are worried about a second wave coming when they open the schools, and if the schools are in person versus, on the other hand, getting children to be in a school in a social environment is also important. So what's your opinion on that?
I mean, I think with modified regulations in communities that have low numbers like we do now, we have pretty good numbers now.
Mm-hmm.
I think that the risk is justifiable. You know, you need to very closely monitor. In an ideal situation, you would test the kids every Friday and only let the kids go back if they're okay. We would have to get better tests. I mean, I already got tested like 3 times, and getting these nasopharyngeal tests to do this to children every week is going to be difficult.
Mm.
But if we get a nasal test, that's what they do at the Broad Institute. That's much easier to do. And if we then sort of manage to just test the entire class in one PCR reaction and only open it up if it's positive, that would be one option. In Europe, they have started going back to school, and they have been able to contain. The teachers are scared, I understand. A lot of teachers are old. The teachers can protect themselves. They can wear an N95, it's not great. They can teach behind plexiglass. You know, healthcare providers have to go to work. I wear an N95 every day. I teach behind a mask. I think the risk is going to be calculable, and you know, we just have to have very strict cutoffs when we shut down the schools.
How about the option of a serological test as sort of a screening, and then going to the nasal test as a
I think the serological test doesn't help us at all.
Okay.
It only helps us to gauge how many people in the community are infected, but it doesn't help at all. The only thing that helps is PCR testing.
Mm-hmm.
And symptom testing. But you know, among kids, you are going to have a higher number of kids that don't have any symptoms at all that are positive. The kids, the smaller kids, their circle is not that big. You know, the college kids that go to bars and get drunk and stuff like that, that's a different story. But I mean, I think we need to understand, you know, this is not going to go away. And even if we start vaccinating tomorrow, we're going to have to deal with this pandemic for some time still.
Right.
So we need to learn to live with it and be safe. And, you know, you need to cut down on crowd size. That's the most important. And distance is important. I think I just flew to Germany, and, you know, I saw tons of little kids on the plane. And they wear masks, they do okay with it. Mickey Mouse makes masks. I think it's exaggerated how cumbersome this is for kids. I think kids can learn to run around with a mask, and you know, we need to get our testing capability up.
Okay, thank you very much. I really appreciate your taking the time to do this. It's been an extremely informative lecture, and I think everybody can agree that we really enjoyed it. So thank you very much. I appreciate it.
Okay, bye-bye.
Bye.
Thank you. Bye.